As mentioned, my name is Kiet Lee, and I'm the founder and CEO of TANA Therapeutics. And I'm super excited to be here and to share with you a little bit about our biotech startup. So we have a fantastic team of seasoned founders and entrepreneurs, a stellar basic scientists and clinician scientists group. But most importantly is that we firmly believe that TANA can accelerate and transform the new frontier of medicine with our intracellular drug delivery tool, which we call TANA Porter. Now,
how do you spell that?
TANA Porter.
TANA Porter.
TANA, TANA, T-A-N-A, and Porter as in Porter, a carrier, a delivery type. So essentially, Moderna, Pfizer, Johnson & Johnson, we all know them and know them for their DNA and mRNA COVID vaccines. And they're part of an exciting new frontier of medicine, which is cell reprogramming or gene editing. In essence, we're talking about tissue engineering, and that's hundreds of billions of dollars in market value. What people don't really know is that these are large proteins that can't get into the cells by themselves. And so that creates a big problem, but a tremendous need and a multi-billion-dollar opportunity for companies that can develop an effective and efficient intracellular delivery tool. Delivery tools, there are three main problems. Essentially, what we're looking at is that our cell is built to keep foreign materials out. And they do take them in, but they take them in to prepare them for digestion. And this process is known as the endocytic defense pathway. So essentially, the cells form these vacuoles that you can see on the slide. And as those vacuoles mature, they digest whatever the content is within. Now, most delivery tools, such as lipid nanoparticles or viral vectors or cell-penetrating peptides like ours, utilizes this pathway to make deliveries. And the products that are available in the market right now are inefficient at one of three critical bottlenecks. The first bottleneck is that they're unable to stimulate endosome formation, so therefore they can't transport whatever payload that is outside the cell into the cell. The second problem is that even if they were able to transport it, once they couldn't, they're inefficient in stimulating the endosome to release the cargo so that it can go into the cytosol or inside the cell and move to where it needs to be. And then the final problem is toxicity, that they disrupt the cell too much that leads to cell death. And this is depicted by the micrograph that you can see in image 3, where you see all these green dots. The green dots actually depict dead cells. This is about 50% of the cells that died. So in essence, what we have developed is an intracellular delivery tool that is 90% effective in delivery and release, as you can see in this in one of the micrographs that we have here. What we have been able to demonstrate is that we can deliver small molecules, we can deliver nuclear proteins, nanoparticles, and then we have also delivered in live mouse models a gene editing called Cre-recombinase. What we're looking for in terms of, so we're actually in our seed round right now, and we're seeking $2 million to essentially do more animal studies, as well as then to develop our CRO, CDMO work activities, and for the administrative work.